Good. Wonderful. Because otherwise he has to move the camera. Yeah. Yeah. So, also, we start with the second round of our meeting today. And I'm really happy to have you, Thomas, here from the University of Vienna. And you stay a long time in US together with Megan Sykes and take part of the exciting work which was done by the Boston group. And he was able to take over all these experiences and adapted this. And I'm really happy to have you here telling us a little bit more about combination of cell therapy with recipients Treg and bone marrow in HLA mismatched kidney. Yeah, so we are happy. Please start.
Thank you very much for the very kind introduction and also for the very nice invitation. It's really a pleasure for me to be here. As Peter has mentioned, I've been interested for a long time in trying to induce tolerance through mixed chimerism. And as you know, of course, there are several clinical pilot trials that have been published that achieved intentional tolerance induction in kidney recipients through the co-transplantation of donor bone marrow that induces chimerism. But one of the downsides of this approach is that these protocols that have been tested so far always require recipient myelosuppression. So there's some type of irradiation involved of the recipient. And this, for many clinicians, is a big hurdle that this approach can be translated to a larger group of patients. Now, of course, this meeting illustrates the high potential of adoptive Treg therapy with a lot of encouraging results, with safety demonstrated in early clinical trials. But so far, it's at least my understanding, there are no robust data showing that Treg therapy on its own can induce tolerance in kidney transplantation. Over the last years, my group and several others have demonstrated when you combine Treg therapy with donor bone marrow infusion, you can induce chimerism and tolerance without any immunosuppression in mice. And there's also some limited experience from the group of Megan Sykes that this combination of donor bone marrow infusion with Treg therapy can improve chimerism in non-human primates. So a few years ago, we started to translate this approach to the clinical setting and started an academic phase 1/2 single center trial. And this is an endeavor and a project that I'm doing together with Rainer Oberbauer from Vienna and Matthias Edinger from Regensburg. So the basic principle is that we are combining donor bone marrow infusion with in vitro expanded recipient Treg therapy in living donor kidney recipients. We also add a short course of tocilizumab, so IL-6 receptor blockade. This is based on preclinical data showing that this promotes chimerism induction. And one of the key aspects of this protocol is that there is no myelosuppression involved. Now, again, as mentioned, it's a single center trial. We include a control group, a reference group, but this is a non-randomized open label group. The predetermined sample size was 6 patients per group, and this is the enrollment period here. The key inclusion criteria, basically we include living donor kidney recipients with low immunologic risk, so they have to be negative for DSA, they have to receive an ABO compatible kidney transplant, and had to be first time recipients. And there has to be an HLA mismatch between donor and recipient. The predefined co-primary endpoints were safety, of course, in this early phase trial, and then efficacy we defined as the individual peak chimerism levels within the first month post-transplant. Now, here in a bit more detail, the treatment protocol, it starts already 2 weeks prior to the scheduled kidney transplant. The recipient undergoes leukapheresis. The cells are transported to Regensburg, and Matthias Edinger and his group there manufacture the Treg product. Immediately after the leukapheresis, the patient receives ATG, so this is typically 2 weeks prior to the transplant. And ATG here is given together with steroids as monotherapy, and then until the time of transplant, the patient doesn't receive any additional immunosuppression. On day 0 then, the donor not only donates the kidney, but also bone marrow. The transplant is performed, and then within the first 3 days post-transplant, the recipient receives the autologous in vitro expanded Tregs, and shortly thereafter the donor bone marrow infusion. The recipient also receives 4 doses of tocilizumab within the first 3 weeks. The baseline, the concomitant immunosuppression consists of belatacept, rapamycin, and steroids. If after 6 months the surveillance biopsy is clear and there are no DSA and the patient is doing well, then steroids and rapamycin are gradually withdrawn, so that the goal after 1 year is that the patient receives only belatacept. So belatacept monotherapy is the goal, which of course means it's not a true tolerance trial, it's basically a minimization trial. So as mentioned, there is a control group. The differences from the treatment group are listed here. Control group doesn't receive any Tregs, no bone marrow, no tocilizumab, and the thymoglobulin starts on day 0, as is typically done in kidney transplants, and there's no scheduled immunosuppression reduction. Now, this is the patient disposition. Over about 4 years, we screened or pre-screened 88 donor-recipient pairs. Eventually, we're able to enroll and treat 6 patients in both groups, as was the target. 1 patient in the control group withdrew consent 3 months post-transplant, so 5 patients in the control group finished the follow-up of 12 months, and all patients in the Treg therapy group. I should also mention 1 patient that was originally enrolled in the treatment group could not be treated because the cell product was out of specification, and so the patient was withdrawn. So 6 patients per group, shown here very briefly. As mentioned, there had to be at least 1 HLA mismatch, but you can see the HLA mismatch spans the whole, almost the whole range, from 1 single mismatch to 9 out of 10 mismatches. Half of the patients in both groups were living related and half unrelated. Now, all 6 patients were treated with both cell products. 4 of the 6 patients received the maximum allowed Treg dose of 15 million cells per kilogram. The other 2 patients received about 10 million Treg cells per kilogram. In addition, the patients received the donor bone marrow cells. The cell number of the donor bone marrow cells was somewhat below in most patients than what we were targeting for, but all patients received both cell products without any immediate adverse events, so the 2 infusions were well tolerated. Now, just very briefly, the individual clinical courses of the 6 study group patients. First patient, as I think is many times the case, had the best course, a completely uneventful course. The patient now finished a 5-year follow-up and is maintained on belatacept. That is actually now spaced every 8 weeks, so belatacept monotherapy every 8 weeks. All surveillance biopsies were clear. The second patient also had a relatively good course and is now 4 years post-transplant, also reached belatacept monotherapy, but his course was remarkable for the development of an isolated DSA already occurring 12 months post-transplant, but all surveillance biopsies were clear. The patient also didn't have any cell-free DNA, but has this single DQ DSA antibody. Third patient, unfortunately, developed a rejection episode during the period when rapamycin and steroids were withdrawn, so triple therapy was reinstated. He's now on dual therapy. The fourth patient also developed a rejection, also developed a rejection episode early on, had for various reasons, also medical and surgical reasons, had a bumpy immediate post-transplant course, but is now 3 years post-transplant doing well on dual therapy. Fifth patient had, unfortunately, complications related to the surveillance biopsy, but immunologically is doing fine and is also now on monotherapy. And the last and sixth patient developed an early rejection and therefore was switched to triple therapy with tacrolimus. Now, as mentioned, one of the unusual aspects of the treatment protocol is that we administered thymoglobulin 2 weeks prior to the transplant as monotherapy. This had various consequences, some of them beneficial, some not. With regard to the cell therapy, the good consequence was that the blood levels, the serum levels of ATG dropped very quickly, so pharmacokinetics were accelerated, so that after 2 to 3 weeks, the Treg specific ATG levels were low enough to administer the cell product without depleting the cell product. On the other hand, of course, this was accompanied by the development of anti-ATG titers very rapidly in all patients, and this led to serum sickness in 5 out of the 7 treated patients. Serum sickness was self-limited without any consequences, but nevertheless is a morbidity associated with this protocol of ATG treatment. Another consequence was not only were the pharmacokinetics accelerated, but also the repopulation kinetics of the lymphocytes were accelerated. As you can see here on the left side, lymphocyte recovery in the study group compared to the control group. And as you can see in the study group, most patients quite rapidly recovered lymphocytes, much more rapidly than when you receive concomitant immunosuppression. And this goes both for the CD4 and the CD8 T cells. One aspect we were interested in was whether there is any indication of clonal deletion in this bone marrow Treg recipients, because as you know, in the experimental setting, clonal deletion is one of the main mechanisms of chimerism-based tolerance. So the group of Rainer Oberbauer tracked TCR repertoires in these patients, and their data show that there is indication, especially in the CD4 compartment, that in the study group, donor-reactive clones are reduced below the level of baseline. So there is some indication that partial clonal deletion of donor-reactive CD4 T cells occurs in these patients. Now, finally, as mentioned, chimerism was one of our main endpoints, of course. We measured chimerism by PCR at 5 predefined time points within the first month. And as defined, we compared then the individual peak chimerism levels as we have predefined. And as you can see, in none of the 6 control group patients, any chimerism was detected by this very sensitive technology. So the detection limit for chimerism is 0.1%. So none of the 6 control group patients showed any chimerism in the first month, whereas all 6 patients in the study group showed chimerism. Of course, chimerism levels, this is total leukocyte chimerism. Chimerism levels are very low, but please keep in mind these are patients that didn't receive any cytotoxic drugs or any irradiation. Now, we also looked at lineage-specific chimerism. This is also PCR data for CD4 and CD8 T cells, because in preclinical work, it has been shown that donor T cell, donor lymphocyte chimerism correlates empirically very well with tolerance induction. So we were interested in T cell donor T cell chimerism. As you can see, the 6 patients developed low levels, and some of them a bit higher levels of donor T cell chimerism, both in the CD4 and the CD8 lineage. Of course, also this is relatively low compared to protocols where you give irradiation and cytotoxic drugs. So we wanted, of course, to check whether these are really nice donor T cell population. And in some patients at some time points, we were able to check chimerism also by flow cytometry. You need the right antibody combinations. But one example is shown here. You can see anti-donor HLA versus anti-recipient HLA. And here in this example, you can identify a donor CD4 T cell population and also a donor CD8 T cell population. So we are confident that these are real donor T cell chimerism levels. So to summarize, this pilot trial indicates that combined therapy with Tregs and donor bone marrow is feasible and safe. Belatacept monotherapy was reached in 3 out of 6 patients so far, dual therapy in the other 2 patients, but we also saw T-cell mediated rejection in 3 patients. We didn't see any antibody-mediated rejection. Total chimerism and lineage-specific chimerism were induced in the study group without myelosuppression, although, of course, at low levels. And with this, I would like to thank everyone who contributed to this trial, and I'd like to thank you for your attention.
Yes, thank you, Thomas, again, for showing the data. It is, let's say, a very ambitious work to do such a trial. In particular, I know Oberbauer very well, to convince, let's say, the physician to come up with this kind of combination, because if I remember correctly, Megan start with a more or less well-defined indication that the patient had malignancy, and the bone marrow transplantation was, of course, necessary. And so here the topic is a little bit different. What was the statement of, also at the end you achieve it, but do you have anticipated ethical concerns in the discussion with your ethic commission, but also with the patient? I think we have patient representatives here, and I think they are interested in to hear what you experienced.
Yes, no, that is a very important question. I have to say, in the defense of the nephrologists, it was not Rainer Oberbauer who was the hardest to convince of this protocol.
No, I know.
But in all seriousness, of course, you're absolutely right. The ethical issues and the safety issues come first. And so we had extensive discussions with many people involved, also, of course, some international experts we knew and so on. So what is the risk of chimerism-based tolerance protocols? Of course, HLA mismatched bone marrow transplantation has a risk of graft versus host disease. I think this is on everyone's mind, and this is a complication so serious that you have to avoid it in living donor kidney transplantation.
Exactly.
And everyone in Vienna on the team agreed with this 100%. So that was a top priority. But since we're not giving myelosuppression, actually the risk decreased, because the question was, with this protocol, without irradiation, without cytotoxic drugs, would we see any chimerism at all? And as I mentioned, the chimerism is low. There's no good evidence in the literature how much chimerism you need, and it probably varies with the protocol details. So since chimerism was very low, and since it disappears, with the disappearance of the donor T cells, the risk of graft versus host disease also disappears. So that was our top priority, to have basically 0% risk of graft versus host disease. Also no irradiation, no cytotoxic drugs. So I think we took a lot of effort to avoid any significant risk to the patients. And most patients, sorry, because you asked how the patients reacted. Almost every patient that we approached wanted to do this.
Okay.
We had 2 patients, for time reasons I skipped this, we had 2 patients which we wanted to enroll in the study group, but then we had to test whether the donor has antibodies versus the recipient. So recipient-specific antibodies, because this is a risk for the bone marrow transplant. And 2 patients actually had these recipient-specific antibodies, and then we had to tell them we have to withdraw them from the study group. And both of them were disappointed.
Okay.
So this is just because you ask about the patient perspective.
Very nice. I have a question to to the stem cell product, is it depleted of T cell because the IgG is not longer present if you give the normally the IgG do the job, but did you deplete the?
That's also a very important question. We used unseparated bone marrow, so we did not deplete it, and this was based on the Boston experience that you you mentioned. Of course, our protocol is very different, but in some aspects, nevertheless, similar to what has been done in Boston. So that's why we opted for unseparated bone marrow cells.
Well, that's surprising that you don't see GVH.
Yes.
Well,
yes.
Well, we discussed this extensively also with the hematologist. I mean, we don't. The patient has a basically unchanged T cell repertoire and hematopoietic repertoire. And in this case, you reduce the risk of graft-versus-host disease, of course.
And regarding belatacept, are you not afraid that they block the Treg?
Yes, I'm afraid. I am afraid, but we had two choices, also for safety reasons. We had two choices, belatacept-based regimen or tacrolimus-based regimen. And I think there are also tacrolimus-based regimens are not ideal. As I understand, that's why you engineer those Tregs that are resistant to tacrolimus. So we had only those two choices. And on balance, I thought belatacept would be more attractive also because there's a lot of preclinical experimental data on CTLA-4 IgE promoting bone marrow engraftment and so on. But of course, you're absolutely right. It's not the ideal drug, definitely not.
Okay. Further questions? No any. Then I have to thank again, and we continue with you, Fadi, and the results, or let's say not really the results, because they couldn't be there so far. Some of the two study, transplantation without over-immune suppression is a name, and okay.
Thank you very much, Peter. I mean, the name, by the way, transplantation without over-immune suppression, that came afterwards because we had the one study, and then what do we call the next one? We have no imagination, so we called the 2 study, and then we said, well, what does 2 stand for? And it's that. So, you know, just to be clear, because I know Antonia doesn't quite like the over-immunosuppression. Okay, we got that out of the way. So this is essentially the protocol that we use for polyclonal Treg therapy in clinical transplantation. So patients have a unit of blood. We take just blood. We don't do a leukapheresis product, around 370 mils, and that's then taken to a GMP unit where the Tregs are selected in magnetic bead-based method, expanded ex vivo, QC'd, and then returned to the patient. And the way that we do the expansion is through this protocol, which is very simple: volume depletion, CD8 depletion, then CD25 enrichment with Miltenyi Clinimax, and then expansion over 3 rounds with TCR stimulation, IL-2 and rapamycin. And then we cryopreserve the final product, so you can leave it frozen to infuse at a later date. And we have up to around 2-year data of freezing to show that the cell product at that stage is still viable. And then the standard QC for the cell product is looking at phenotype. Does it express FOXP3? Have the beads been removed? Is it viable? Is it potent? By that, that's a suppression assay, and I'll show you some results from that. And is it sterile? So the first few things I want to show you are just about the production of the cell product, because it's not easy, and there are failures. And there are failures that sometimes are to do with Treg expansion. So this here shows you just a few of the cell products that we've produced in GMP, and you can see that there's basically no relation between the starting lymphocyte count or the white cell count of the patient and how many Tregs you have at the end. It's not relevant. What appears to be more relevant is perhaps the phenotype of the Tregs that go into the product. So there is some hint that if there's more naive Tregs that go into the starting product, you're more likely to expand cells. And then when you look at the in vitro function of these cells at the end, so these are actually 64 clinical grade batches that we've produced. This is suppression assays with 1:1, 1:5, or 1:10 of Tregs to stimulators to no Treg stimulators. You can see that most of them pass, but there are actually a few that don't pass the QC of 60% suppression. So we have failures both at the suppression side, we have failures in cell expansion, and we have the occasional contamination as well, which as you know, is a problem with cell production. So I just wanted to give you an overview of how the process works. It's not always that easy. We've looked at phenotyping these cells here using CytSeq, and on the left you can see the TR001 cell product. These are the cells that we make. On the right are the Tregs in the peripheral blood of the patient, and you can see they actually look quite different after expansion compared to the cells in the patient. Apart from the migratory cells here, these we term them migratory because of some of the things they express, but those actually include cells that are both in peripheral blood and the expanded product. If you look at what they're expressing, they generally express FOXP3 RNA quite well. They express CD25, both at the RNA and the protein level, and they are not naive after expansion compared to the peripheral blood Tregs. In terms of their expression of Helios, some of them express Helios here, so the purple on the left is positive for FOXP3 and Helios, so about 20%, but actually a lot of them don't express Helios. And then looking at their proliferative capacity, so this is using a proliferation score based on the gene expression. There is a population within them that appear proliferative down here, but the majority are not. The majority look like the rest of the peripheral blood Tregs. And finally, when you zoom in on that CytSeq population to see actually what's in it, in just the TR001 product, you can see that there are actually lots of different Treg populations in there. Now, I think this is partly a function of, you keep zooming in, you'll keep creating clusters, that's the way that it works, but it's not a uniform population. There are lots of different things within it. Perhaps that's not surprising. But when you look in more detail at all those different populations that I've just shown you on the UMAP, they all express a core Treg module. So they all express the things that you would expect Tregs to express. And so they look like Tregs, they function like Tregs in suppressive assays, so they probably are Tregs. So I'm just now going to go into some of the clinical data, but to do that, I have to just briefly talk about the one study, because it was the prelude to where we got to with the two studies. So the one study was before ReSHAPE, and here we treated 12 patients with Tregs in a dose escalation trial in the UK. And we now, believe it or not, have 10-year follow-up data for one study patients. It's been that long, yeah. So you can see here that the cell therapy patients in green and the control patients in blue have done very well in terms of both their patient and graft survival. There have been a few graft losses in the control patients, but that is not unexpected at this stage. We're 10 years post-transplantation, so it's within the normal range. But it's actually quite reassuring that the cell therapy patients continue to do very well. In terms of the renal function, they, again, generally doing well in terms of EGFR. The cell therapy patients slightly better than the control patients. And you can see that's due to some changes that happened from 15 months onwards in the control patients. In terms of their immunosuppressive burden, here you can see serum tacrolimus levels are quite similar between the two groups, between the cell therapy and control, so it's not that the cell therapy patients are getting more tacrolimus at this stage, although it is important to note that a few of the cell therapy patients, 4 of the cell therapy patients, have been taken off MMF. So actually their immunosuppressive burden is slightly lower, the cell therapy patients, compared to control, but not in terms of their TAC levels. And then infection rates, again here at 10 years, you can see most of the events, as you expect, happen quite early, but the CMV status, this is percentage seronegativity on the y-axis, is generally the same, perhaps slightly better in the cell therapy group. Okay, so I'm now going to talk about the 2 study and where we went here. So the 2 study, we originally designed this in what we thought was quite an intelligent way, because we wanted to give Tregs quite late, at the time when cells were starting to repopulate after CAM-path induction. And why CAM-path induction? Because in Oxford it was the standard of care, and I understand that it's clearly not the standard of care everywhere else, but in Oxford, CAM-path is what all our transplant patients get. And that was really based on the 3C study, which showed that CAM-path induction in the early stages is better than basiliximab induction, at least in the first 12 months post-transplantation. So we have our Treg arm and a control arm. And in the Treg arm, patients have CAM-path induction day 0. They're maintained then on tacrolimus and MMF. And then at week 12, start to have a slow reduction in MMF, which is stopped at week 26, at which point, a few days later, patients get a dose of regulatory T cells between 5 to 10 million cells per kilogram, which is the dose taken from the 1 study. And then they're maintained on tacrolimus monotherapy. At week 40 onwards, there's an option to also start to reduce tacrolimus monotherapy, although not necessarily stop it. The control arm have standard of care immunosuppression, so CAM-path induction followed by tacrolimus and MMF, normal. The red triangles here are important because those are biopsies. So we have biopsies before infusion of Tregs and after infusion of Tregs, and a biopsy that lines up with the control group at week 38. This is important because in Oxford we don't do protocol biopsies as standard of care, so this is only part of the trial. This is a randomized trial, so patients come and they get randomized to a group and they get one of them. It's a challenge, and I think, for future trials, it's something people need to think about when doing transplant trials, whether they really need to be truly randomized. In terms of the clinical outcomes from this trial, and I'm only talking about now this trial, because this trial was then stopped during COVID, because CAM-path was seen as not a good thing to be given during a global pandemic. So that trial stopped after we recruited and randomized 7 patients to the trial. And now I'm showing you those 7 patients here. So we have here the cell therapy group in red and the control patients in blue. Generally very similar across the course of the follow-up period. Here, this is looking in weeks. And apart from one episode here in one of the control patients of tacrolimus toxicity, which then resolved. But generally, the patients did well in terms of creatinine levels, GFR, and tacrolimus levels, as you can see here. That's really most of what we can say about the 2 study clinical outcomes up till now, because we've had no rejections. The patients are all doing well in either arm. The patients who have stopped MMF did not reject. And tacrolimus has been slowly reduced in the cell therapy patients, and they're doing okay. So I think it's in that very small cohort, it's shown us that you can give Tregs quite late after transplantation, that it's safe to do it, and it has had no impact on their clinical outcomes. So I think it's a good trial of late infusion in the presence of this depleting induction immunosuppression. We've looked at Treg survival in the patients, and you can see here that after you infuse Tregs here between weeks 26 to 27, there's perhaps a slight increase in the number of Tregs in the peripheral blood in the patients who have it. We don't actually have a measure in the control patients at that time, so we don't know where their Tregs are. There may be, the lines may not be exactly where they are, but there is perhaps a slight increase. And this has also been shown in the 1 study, that you give Tregs and you can see Tregs at least early on. Later on becomes less easy to detect them in the peripheral blood. We did, however, see that after you have Treg therapy here, that there is a slight increase in the number of transitional B cells in the peripheral blood. And again, this is something that there were hints of in the one study cohorts. And if you look here in those patients using, again, single cell sequencing of the peripheral blood, you can see the cell therapy on the right, the point of cell therapy infusion, the green arrow. There is a slight increase in the yellow box I've just highlighted in Tregs after you infuse the Tregs, but there is slightly later on at week 49 a huge increase in B cells in the peripheral blood by single cell sequencing. So this is something that we're still looking into, and it's not mirrored in the control group. So there's something happening specifically in the B cell populations. And then we've looked at the biopsies of these patients. So in fact, originally, even in the one study, it was noted that the patients who had Tregs had these very dense, focal rather, infiltrates in the protocol biopsies, and that these looked very much CD4 positive with some FOXP3. And at the time, they were worrying, but the patients were doing well clinically, and so they were thought to be non-consequential, and patients continued to reduce their immunosuppression. But we've started to look in more detail in our 2 study biopsies and also some of the 1 study biopsies to see what's happening in them. And the first thing to say is if you do single cell sequencing of the biopsies from these patients, you get more cells back if they've had Tregs compared to control. This is single cell sequencing of CD45 positive cells. So we take a biopsy, we dissociate, sort the CD45 cells, and then you can see actually there are more cells in general if the patients have had Tregs. And then when you look by spatial transcriptomic approaches here using geomics profiling, you can actually delve deeper into what these cells could be. So here I'm showing you cell therapy patients on the left and some rejection biopsies on the right, control rejection biopsies on the right. And what we did was highlight areas in the biopsies of these infiltrates and try and see what their makeup is. So on PCA, using this geomics profiling on the left, you can see we have 3 populations that separate quite well. There are patients in green who are the cell therapy patients. Blue is rejection. And these orange ones, those are no infiltrate in the cell therapy. So we took areas within the cell therapy biopsies without an infiltrate, and they're different as well. So I think the first thing to say is that infiltrates in rejection compared to cell therapy patients are not the same. They may look worrying, but they don't look similar in terms of their expression profile. And the things that appear to be contributing to those differences on here, again, on the PCA, the major components are things like CD20, so B cells, Ki67, a proliferation marker, some macrophage monocyte markers, and then some cytotoxic cellular markers as well. And if you look in a bit more detail at that, we have here on the green, we have the cell therapy patients. You can see that they have a different expression to the rejection patients. And in particular, you do see things like CD20 and FOXP3 highlighted in the patients who've had cell therapy in those infiltrates by spatial profiling. We've also then looked to see whether some of this can be confirmed by immunofluorescence staining, and CD20 was one of the things that is increased by IF at the protein level. And then if you just delve a bit more deeper in terms of what the FOXP3 positive cells are expressing compared to the CD4 positive cells within an infiltrate, both in cell therapy and rejection. So this is, again, spatial profiling, but now we're creating a mask around FOXP3 positive cells to see what they express, and a mask around CD4 positive cells that are FOXP3 negative to see what they express, and they're different. So those cells look like Tregs. They very highly express FOXP3. And in terms of their RNA profiling spatially, again, this confirms some of the results from the protein profiling. So you have a different program in cell therapy compared to rejection. So you can see here, in terms of rejection, you have those things that you typically might see, and particularly CXCL9 and 10, those gamma-induced chemokine ligands, and also you have in the cell therapy IL-10, Helios, TIGIT, etc. coming up. So again, just confirming that they look very different. And then if we look at single cell sequencing of the biopsies here, assessing particularly the cell states of the cells that are infiltrating. And I just want to highlight, first of all, a Treg cell state here in the top left, but also down here in the cell therapy patients here, this very specific B cell state that is appearing in patients who have cell therapy, which looks like marginal zone B1 cells in the biopsies. So we've also then looked at single cell spatial profiling in the biopsy. So now what I showed you before was geomics, which is bulk sequencing of regions. I've shown you single cell sequencing, and now I'm showing you single cell in situ sequencing of the biopsies. And this is just showing you the kind of resolution we can get. And here is, to be honest, I'm actually just showing this to show off. I haven't analyzed the data, so it's just nice images. So this here is a biopsy from one of the cell therapy patients. And we've clustered the cells using the single cell in situ sequencing by color, according to what they are. And you can see that there are infiltrates here highlighted. And if you look in more detail at those infiltrates and what cells are within them, there are a few T cells. There are lots of cells that express Helios, and there's some cells that express FOXP3. So perhaps there are some Tregs also getting into those infiltrates. And then if you look at every single infiltrate in those cell therapy biopsies, we wondered whether they were different. Were they temporally different? Were some more advanced than others? Were some more rejection looking? Were some more regulation looking infiltrates? And actually, interestingly, they all look very similar. So these are highlighted, some of the infiltrates in the cell therapy patients. And in terms of their makeup, here the gray, blue, purple, and yellow, the makeup of the cells that's within them looks quite similar. So it doesn't look like some are more worrying than others. We've also started to look at the cellular interactions in there, because what's the point of doing spatial if you can't see what the cells are communicating with? And we have started to look at this, and you can see that some things are coming up here. For example, T cells interacting with B cells. So perhaps there's something going on in those infiltrates there. So that's the first bit of the two study. The second bit of the two study, after we had to stop it because of COVID, actually reverted to the one study protocol. So we went back to basiliximab induction in the control arm, and no induction in the regulatory T cell arm, and an early dose of Tregs instead. So this is the trial that's currently running in terms of the two studies. So again, randomized controlled. The control arm have basiliximab induction and standard immunosuppression. The Treg arm have blood procured. They have no induction immunosuppression, and at day 5 they have a dose of Tregs, and then they have their MMF reduced and similar biopsy time points as the original design. So we're still recruiting to that trial, and as it stands, we have 48 patients recruited in the trial. So that's pretty significant for a Treg cell therapy trial in living donor kidney transplantation, and we're aiming to get to 68. So I hope by this time next year we'll have completed it. So that's all I have to say. Thank you very much for your attention. Happy to take questions.
You are, as always, great.
Thank you.
So, Abul. Yeah.
Couple of questions. That's a heroic endeavor. So you should be congratulated.
Yeah.
But given that these are small numbers, can you say that the Tregs are doing something or not doing anything?
On a clinical level,
They do.
yeah, in a clinical level, look, it is difficult to say. I agree, it's small numbers. But the patients who've had cell therapy are coming off immunosuppression slowly. I mean, they're not off completely, and they haven't had rejection episodes. So I think that, combined with the fact that they seem to have fewer infection episodes, they haven't had to have more immunosuppression, or, at any point, and a few of them have had no induction immunosuppression as well. I think that is very good. Now, is it actually the Tregs, or are we just very bad at immunosuppressing patients, and patients should just be having less immunosuppression? Maybe, right? It's possible, and maybe this is giving us the impetus to reduce immunosuppression when we should be doing that anyway with patients.
Yeah.
Right? I don't know.
But there's a follow-up. I have a follow-up to that question, which is, in order to see the effects of Tregs, if any, and we'll leave that as an open question, maybe we are not doing the right assays. Maybe we should be doing something else. I mean, go back to old-fashioned donor-specific MLRs.
No, that absolutely, we should, and we are doing those. I think that is important. But again, it's just this functional readout of what you still want something clinical. I actually think the biopsies are super interesting. And I think we, in general, are very bad at analyzing biopsies. The Banff score is poor.
Exactly.
It's just so broad. It doesn't give you any granularity about what's happening. I think we need to be able to understand more what's happening locally. So
Eons ago, I remember Suthan and people like that had this story that in some tolerance protocol, I don't remember the protocol, when they looked at urine RNA, the signal that jumped out was B cells.
yeah, I mean, B cells is a theme. B cells was a theme in the ITN project, so it was one of the signals that came out of the operationally tolerant patients, right? It's coming out in some experiments in Pittsburgh, the Pittsburgh group who are doing mouse-to-mouse experiments of kind of spontaneous tolerance in kidneys, a specific kind of match. B cells are what is coming out in the kidneys. So there are lots of things, that seem to be indicating that B cells are relevant. Now, are they causative or associative? I don't know. But maybe if you do a biopsy in a patient and you see B cells, maybe that might make you more likely to take immunosuppression off a bit. Maybe.
Dirk?
Thank you, very nice presentation. For the batches that you made, the 64 batches, you saw a difference in the expansion capacity and also suppression capacity. Did you look at the effect of immunosenescence markers on those results?
No, I think so. Not specifically, but we have all the single cell data from those. Well, not all the batches. We have of a subsection of the. We have the single cell data, so we can look and see what's happening in those particular pathways. In case that might be contributing to the lack of expansion, it's possible. Yeah, thank you for that. Thank you.
Robert?
Sorry, I'm actually having two completely differing questions. Question number one, I really was impressed by the huge number of cells in the Treg group, which is a kind of scary, because one would consider the opposite effect.
That
Whatever those cells are, is there any experimental or translational data that is comparable and would explain what you are seeing? And
so you're asking about the number of cells that are infiltrating the kidneys.
The imaging.
Yeah, I think so. I think the thing that needs to be taken away is that they still don't look like rejection biopsies, but if you just take a Banff score and you say, oh, look, there's cells, okay, that doesn't look good. But if you actually look at what they're made up of, they look completely different to rejection biopsies. I think that's the key. And then there is this, as I say, this experimental evidence from Pittsburgh about kidney biopsies and tolerance, and they have infiltrates that look quite similar. And then if you look back, if you go back, decades, the Quilty effect in heart transplants, that was similarly, they had these infiltrates, but no rejection. So it's not, I think we've forgotten a lot of these, that actually not all T cells are bad.
Which is fascinating. The other question I would like to ask, just from a practical point of view, as far as you have quite a good number of patients enrolled in the study, what was the, or what is the dropout rate concerning technical problems in producing or
it's a very, very good
enlarging
question.
the cell pool?
Yeah, so in terms of Treg cell production, we have around perhaps a 15% failure to produce rate. Some of those patients have another go at expansion, and then they're okay. Some of them, there's no time because their transplant has been dated. You've taken blood in advance, and they don't want to move their transplant, which is absolutely fair. So that's the rate that we're working with at the moment. I think it's not good enough, and I think the process actually needs to be massively improved. And I'm submitting grants to do that. And if any of you review those grants, please let them through.
I will.
Yeah. Okay, thank you. Yeah. So that, the production process. If we want to make this, if we want to go into a phase 3, there's no chance with this production process.
Thanks. Micha?
Seeking for hints of Treg efficiency, do you see clonal expansion of the Tregs in the?
No, interestingly, so we've looked at the clones of the Tregs post-expansion, and it's very polyclonal. We don't see
TCR profiling in the tissues.
And then we've looked at so it's 5' sequencing that we've done in the tissues, and there's not a huge amount of overlap between the Tregs and what we see there in the tissues, but I think it's because it's very polyclonal. I wish there was a few clones expanding that then you could detect, but that's not the case. Similarly in the peripheral blood.
Yeah. That is a difference to what we have observed.
Yeah,
we saw really over the time of I think 1 year, we saw really a shift towards clonal expansion, and we think this is pretty good, but nevertheless,
difficult to know.
So, thank you, Fadi, again. Great.
Ein Moment. We still have one question.
Okay, sorry.
Yeah,
Sorry, I haven't seen it.
Actually, I have a technical question regarding the suppression of the Tregs, and I see you have really good suppression for the Tregs, and may I ask if it is autologous setting or?
It's autologous, autologous bead stimulated.
Okay, thanks. And also for the survival of the treatment group versus control group, and the survival rate is related to the age or some other?
In the control group where there have been transplant losses, I don't think there was anything specific that meant that those patients lost their grafts, but it is 10 years, so it happens.
Okay, thank you.
Yeah, thank you.
Thank you, Fadi. So, can I have my slide, please? No? That's not me. This is, I think, Carol, right? Oh, who is it? No, it's Piotr. Piotr, then you. Yes, you. He put. Huh? It's. I know it, but please, I need the right presentation. Otherwise, Piotr, you can take it over. I'm very open. This is a ProTrack. ProTrack trial, please. Yes, it is. So I was not so smart to present the data we have generated also in the one study. We have, I think, now data from 7 years, and I can tell you that even with our approach, based on the one study protocol, all patients who have achieved at 1 year after transplantation a monotherapy with very low dose, tacrolimus, DRAF level below 5, stay for 7 years with excellent graft function on monotherapy. It is also not a tolerance induction, and we have the same observation. But again, what we want to introduce now is that we ask ourselves, are we able to translate the data coming out from the living transplant setting towards more difficult, let's say, transplant population, and this is a diseased transplant population. And I will shortly mention what is let's say, the main difference with respect to diseased transplant patients. And in any case, we have predetermined recipient factors. They are maybe the same between living and disease transplant patients regarding alloimmunity, pre-existing disease, and so. But we have a significant difference regarding the donor factors with respect to what we call in nephrology the marginal organs, regarding the pre-existence of the disease, which leads to the death, which leads to brain death, and all the cytokine storm, which damage at the end all organs. Also, we have to take into account that ischemia reperfusion injury is a more prominent issue in comparison to the living transplant setting. And so overarching problem what these kidneys, if they get transplanted, have to face is inflammation in the transplanted or in the organ which has to be transplanted. So, and this impact, of course, this impact, of course, what's happened? Of course, the outcome of the graft, and that is the reason why we need immunosuppression. And the point is here a nice study published a few years ago showing us one crucial point, and this is that we lose the grafts or the patient with functional graft due to rejection in roughly 20%. And the rest of complication after solid organ transplantation, and it doesn't matter if we speak about heart, kidney, liver, lung, is related to either cancer or to infection problems or toxicity of the immunosuppressive drugs given to non-transplanted organs or to a higher incidence of cardiovascular events, which means we have to think how we can lowering the burden of immunosuppression long-termly. And here, in particular, with the background of the diseased kidney transplant setting in which the organ is more inflamed, we have to answer the question, can we achieve with regulatory T cells here the same amount of reduction? I will here not go through in detail because everybody knows that regulatory T cells play an important role in interaction with a lot of cells, not only the effector cells, also NK, also B, and also play a role in the differentiation of monocytes towards M2. they secrete a lot of factors which important to long-termly downsize the activity of the other cells. They interact on the metabolic pathways and they modulate, which is one important issue, different antigen-presenting cells like B cells or even like dendritic cells. So the point is what we already discussed in the early morning is if we speak about regulatory T cell therapies, we have to be aware of that we have to figure out what is the right dosage, including in the context also with a combination of different immunosuppressive strategies. Fadi already pointed out, and we are also interested in what is the right time for giving the cells. The production process indeed play an important role about the robustness of the product and also the biologic activity. The product composition, what was excellent demonstrated by Fadi, is not a homogeneous one. Yeah, so therefore we a little bit different from what the colleagues in King's, I think, is the cell producer, right? In King's are doing. We developed from the beginning on already in the one study a process allowing us to use only 50 ml blood, select the cells of interest, expand them, and have a product ready now within 16 to 17 days. We, as I already mentioned, we saw, if we follow up the patient, we saw an excellent safety profile, like Fadi was outlined the results from King's and Oxford, and we see also long-termly with respect to UABOL, we see hints of efficacy also long-termly. Not in all patients, again, particularly not in them who have trouble already at the beginning after transplantation. And this means that we are able to show that we have a stable, that we could achieve in a small cohort, of course, a stable low-dose TAC monotherapy in this patient population. So, but what are the limitations, in particular with respect to the diseased kidney transplant? We saw already in the one study population a temporary increase of the circulating T cells. We have some issues regarding the engraftment of the cells. In this case, it means engraftment or homing to the place of interest. We know that weaning is not successful if you have patients with preformed effector T cells. And again, I already mentioned the question of translatable of results from a kidney transplant which received different kind of, let's say, excellent or not excellent organs. So we are also interested in conditioning the patient, and this is a very old, let's say, experiment we have done in 2012 or even before, in which we could show, at least already at this time, that it is of favor if you have at least enough space for the cells to grow up by any kind of moderate T cell depletion. And This is what we further investigated, because Thomas already pointed out that if you deal with ATG, then you have to be sure that at the end the ATG level is not impacting the survival and functionality of the infused Tregs. But nevertheless, downsizing of recipients, T and B cells, is one important point to allow a better engraftment of the regulatory T cells. It is also favorable, in particular, if we look later to adapt the process towards autoimmunity, that you balance the ratio between regulatory T cells and effector T cells. With ATG, we saw a preferentially depletion of pre-activated T cells, and enhanced and lasting intratissue functionality. With that, we come up with an idea, and that is what I personally have done over a long time in the kidney transplant field, that we use very short and very low ATG use, not what is written in the documents by the company. We come up with a strategy to have 2 times 1.5 milligram per kilogram body weight, and by doing so we investigate what is the half-life if no ATG is more or less present or detectable in the sera, and this is after 40 days, which means if we give the cells in the time frame of 16 to 18 days, which is our production process needed, so then we will not have any problems with the survival and the function of the cells, because in contrast to the living situation, we don't know when we have an organ what we can accept it in place. That means we have to start, we have to come up with a decision, yes and no, and with the production process at the time, if the patient gets a call, come in the clinic, and we have to stand by and organize everything. And therefore it is, let's say, also a matter of not only showing something is translatable, it is also a matter how we have to structure the process to treat the other part of our transplant population appropriately. Also, the trial objective is first to assess whether the administration of the polyclonal autologous Tregs in diseased kidney transplant patients with a temporary lymphopenic situation is durable, is able to modulate the immunological response in such a way that here we can achieve the same what we have seen in the living transplant setting, a monotherapy. Yeah, it's not just a dose reduction, what we have normally, really a monotherapy. And moreover, what we want to see at the end is that this is for a longer period a sustainable achievement. That means that the patient have not only for the time of the study is being a monotherapy in place, that it is a long-lasting effect, even if I personally believe that maybe this patient then later on, 1, 2, 3 years later, need a second shot. So the eligibility criteria is, of course, these patients have to be registered by Eurotransplant for a primary diseased kidney transplantation. We define the age. We define, of course, that they have to have extensively informed, which is also an issue in the small time frame we have between call and transplantation. We decided to test them. At the time, they agree, and just 1 day before they receive the cells, and the fragility of the patient has not been too problematic. So another point here, in contrast also to the living setting, is that we have to define also the donor eligibility criteria. And here one important point is, in particular, at least we here in Germany, we have to face a problem that the donor age get older and older with all the problems. And therefore, if we have a donor, deceased donor, which have more than 2 of the donor factors like hypertension, cerebrovascular accident, or high serum creatine above 1.5 milligram, or a long-term ischemia time, then we say this donor is not eligible. All other procedures, how the transplant surgeon do the surgical procedure, this is not an issue. We will count this, but it is not an issue. So I mentioned already the 2 times very low dose of ATG. And we have a classical 3+3 design. Everybody know it. And we have a control group. And the control group receive, of course, the same induction therapy, also ATG, but then no other things, and stay as it is standard of care on triple drug therapy long-termly. And that is what we decided based on the biomarker monitoring, because induction therapy change some of the biomarker profiles, so we are interested in, and here it is not helpful to look up what we have done in the past in the one study. This is a dose reduction scheme, and you see that we try to come very early to a situation that the patients are free of steroids, that they are free of MMF, and we also tapering, as you see, the draft level for tacrolimus very fast. Let's say to have a draft level lower than 5. The product will be given at day 16 or 17 after transplantation, and we have, of course, 2 major endpoints. One is, of course, related to safety endpoints. It is a phase 1-2A. And here we focus on acute toxicity in case of if we give the product to the patient. One point, of course, is the over-immune suppression, and then the chronic toxicity. And here one important point is that it is at any time an issue if we suppress by regulatory T cells. And by the way, I have to say that we have no hints for the long period observation really regarding a higher risk of malignancy, but nevertheless it is an issue due to the long-term history of the patient, the risk for malignancy in long-term dialysis patient is even higher. So, and therefore we will count this. The clinical endpoint is quite clear. Biopsy confirmed acute rejection within the first 60 weeks after transplantation. Of course, assessed by the Banff criteria, because that is what everybody understands, and all the other things we will do as well. But nevertheless, Banff criteria, if we have an acute rejection, then this is a failure regarding the primary clinical endpoint. And we also heavily interested in chronic rejection because this determines at the end how long we can expect maybe the efficiency of the infused cell product. This is what Dieter already mentioned. We have secondary endpoints defined, clinical one, time to the first acute rejection episode, to the severity, and to the response to treatment, level of total immunosuppression. Are they really on low monotherapy? And prevention of chronic graft dysfunction, I already mentioned. And everything will be accompanied by the biomarker portfolio, which was discussed. And we also look up by a health survey with a standardized quality assessment, the clinical, let's say, improvement which can be achieved. So what are our hurdles? To explain a little bit why we are now just in track, which makes me a little bit upset. And I will explain two hurdles. One is dedicated to the point that in the ones, also first, you see here the situation of many of our patients. They have, due to the long-term time of kidney disease, they have no good veins for any kind of punction. So this is really a critical issue. The second point, they suffer mostly from anemia. It's also a point, therefore we stick on 50 ml. And at one study, it was not an issue, not at all, that we use the diagnostic, let's say, blood collection tube, after we are applying for a clinical trial application, the regulatory authority come up and say, whoa, that is not longer accepted because this blood collection tube is not certified, is only certified for diagnostic only and not pharmaceutical grade. So, by the way, it could be that the heparin contains any animal origin, yeah. So, and the third point was that they really insist that we need for 50 ml blood a certified collection site for coming out with blood for processing the blood for our T-cell product which have to have a manufacturing authorization that we are now able to produce whole blood for this purpose. It was painful because it took us 2.5 years to develop everything in line with the regulation. That means that we had to characterize under GMP compliance the chemical characteristic of the tubes, toxicity, the silicone oil, leak proofness, all the things like that. And this was related to a mess of SOPs, more than 700 pages only for this purpose, beside of 1,800 pages to get this approved, because without that, even if I had the manufacturing authorization for our product, I couldn't produce anything because this was now defined, the peripheral blood was defined as a starting material and not the cells itself. So we achieved it, but it took us time, and therefore we are delayed. And the second important hurdle was and is really that we and some others got the information that Miltony, the beads we normally use, discontinue the production under GMP grade condition, and therefore we had to look up which option we have, and we investigate more or less all of what was available at the market. At the end, we come up with the Dynabeads from Thermo Fisher. So, but here again, yeah, it was not only that we could show these points are comparable. We had to do 3 confirmatory runs with all aspects to get, let's say, an re-approval of what we have already achieved. This includes that we have to provide all information about the Dynabeads antibodies and all the things like that, which was also not nice. And you see here, if you compare the Miltony and the Dynabeads, we have really comparable results, also with respect to the release criteria, and also with respect to all other populations which are in our product included. So, coming up, last but not least, we are successful, could solve all issues. We have a manufacturing pipeline which is stable, which we are more or less really fine. We have a high expansion rate out of the 50 ml with a high viability, high purity, and more or less low impurity in a closed system, in a very rapid production process. We could secure the supply pipeline. We are also working on data for multi-omic single cell for the product specification, like Fadi was showing it. And we have also, after several turns related to what I explained, as a reconfirmation of the IMPD of the manufacturing process, and the trial is accepted. We prepared 10 contracts, and yeah, we are now recruiting the patient. And here I will highlight that I'm really happy and proud, and yeah, that Robert is the PI, Robert Oellinger, and the surgeon are really very helpful in making the trial running. we have Siren in place, and the guys from Shaggy Moon I already highlight. Thank you for your attention. So, do you want to know something?
Thank you. Petra, very, very nice to see that you're overcoming these challenges. What happened with the German authorities? Why did they change? What was the...
Could you ask me a better question, please, which I can answer?
Well, what's their reasoning for these changes?
Fadi, I can. Therefore, sometimes I'm crying a little bit, yeah, and even what now is coming, but the pragmatism, yeah. And the willingness to see the chance of such kind of treatment strategy is not longer in the German DNA, I have to say. Yeah, so this was
German Angst.
German Angst, exactly. This is now like Butterbrot or kindergarten. Yeah, what everybody is understanding. German Angst is indeed an issue. Yeah, and this is of course for academic institutions or for, let's say, developers and persons who are aware of all risks. Yeah, and take the risk. Not the authorities, by the way, take it. It is an issue, yeah, and so it is German Angst. And another question? No. Then it's fine, and we move forward with Laila, showing the life track study, in which we want to discontinue the immunosuppression in liver transplant patients.
Yeah, thank you. So I will present you the background on a phase 1-A open-label single ascending dose study to assess safety and tolerability of regulatory T cells to promote discontinuation of tacrolimus monotherapy in liver transplant recipients. And to understand the background, why we even want to discontinue now the immunosuppression in liver transplantation is basically because as opposed to other organs, liver transplantation or livers are quite tolerogenic. So there's a very low incidence of chronic rejection rates among transplanted liver recipients after 5 years. And actually, after 5 years, or even 10 to 15% of those liver transplanted patients can achieve a state which is called operational tolerance, and means that a complete withdrawal of the immunosuppression can be achieved here. And to give them a chance to maybe achieve the state earlier, we thought that regulatory T cell therapy might be a good idea, and we target here in the specific trial the patients 2 to 3 years after liver transplantation to check whether we can achieve such a state. The clinical trial application is currently under evaluation. We will use the same product as was described already for the ProTrack study. Of course, therefore, we had the same hurdles to overcome here. And the patients who are included in the study should be patients who are already on monotherapy. So these are patients which don't have any additional problems. They are stable on this therapy for around 1 year, and then we want to achieve this complete weaning of the tacrolimus immunosuppression. The primary endpoint here is, of course, also safety regarding acute toxicity and over-immunosuppression. And then as the clinical endpoint, we also chose biopsy-proven acute rejection at week 60 after the Treg infusion. The secondary endpoints regard viral load, HLA-DR, donor-specific antibodies, the TSDR methylation of the Treg in the peripheral blood, then also the clonality, iron metabolism, as this was associated also with the state of operational tolerance, and then excessive leukocyte subset profiling as already introduced in the morning, and then for acute toxicity also acute inflammatory marker release after infusion, so up to 24 hours after infusion we will monitor this. The infusion of the product will be done around 17 days after blood collection, and these patients are longer in the treatment of the outpatient clinic already. So the main interventions which are planned here is first of all the blood collection after the of course informed consent of the patient. Then we need the 17-day period for manufacturing, and in the intervention group we will then administer the Treg product. We will also have a control group which will be matched regarding age and gender, around 3 years of the patients, and then after 30 days we will start to wean the tacrolimus to a lower dose of 3 nanogram per milliliters. So the patients which are included should be on around 5 nanogram per milliliters upon inclusion, and the control group will stay for the whole time on this dose, and then tacrolimus is planned to be discontinued after 60 days completely in the intervention group only, of course, and then we will have a assessment of the primary endpoint after 60 weeks of infusion. We will also have the classical 3+3 design, so we start with a dose of 1 million Treg per kilogram, and then in case we see any dose-limiting toxicities, our group will be put up to 6 patients, and then accordingly up to 3 million Treg per kilogram, and we will have 9 patients in the control group, no matter of dose-limiting toxicities or not. The main inclusion criteria are, of course, liver transplant recipients, which are adult, give their informed consent, then they should be between 24 and 36 months after liver transplantation, at least 6 months stable on tacrolimus monotherapy, and at least 6 months stable liver function, as no greater deviations as 2.5 times from the normal laboratory parameters. And main exclusion criteria are multiple transplants or any kinds of retransplantation, hepatic autoimmunity conditions, malignancies, immune diseases, or any kind of ongoing infections, and of course all ethical required exclusion criteria. I think I've already mentioned the discontinuation scheme quite extensively, so in total we will have 20 trial visits. They will be organized at our outpatient clinic of the surgery, led by Dennis Ulrich, who's PI of the study. We will follow them up to 6 weeks after the Treg-02 infusion in the terms of the clinical trial, and of course then the normal follow-up will still be continued. And besides monitoring of standard laboratory parameters, we will focus on chronic viruses, changing immune cell subsets, and the points I mentioned before and that already have been introduced in the morning. And as I said, directly after infusion, we will measure standard laboratory acute inflammatory marker response by our routine laboratory. So the LiveTrack study was our first experience actually to submit a clinical study in the new CETUS system, which is required by the new legislation of the EU. So here we have now a one-time submission of both the Part 1, which is our classical part, which we were used to submit to the Paul-Ehrlich Institute, so our national authority in Germany, including the main documents such as investigator's brochure, IMPD, and clinical trial protocol. And of course also the second part, which is basically what we used to submit to our ethics committees, which is the patient information and all site suitability documents. And we already have uploaded this after overcoming the problematic of starting material and receiving the official allowances from our departments here at the university, receiving the European clinical trial number, and then our beat problematic came up. This is why we were stopped for roughly 1 year until we could then finally submit successfully our clinical trial application. We received some requests for information regarding formal aspects already in October, which were successfully validated in November. And now, just a few days ago, we received our first request for information regarding the Part 2, so the ethical assessment, which are all things that we can easily solve, and will submit then by next week. And to see where we are in the process, so we are now right in the Part 2 national assessment. We have a few days to submit our additional information now, and then we're waiting for Part 2 assessment, Part 1 assessment, of course. And roughly the whole process should take around 5 months for the initial application. So we expect to have a final decision on our trial in March 25, and then hope to be able to start off already. And as any clinical trial, although we have not recruited yet any kind of patients, and they are maybe the heart of each clinical trial, we already have involved a lot of people behind this, and this includes, of course, the sponsor side, then the GMP facility, the clinics, the study labs, which are both at Charité and also BIH and, of course, Czech Immune. And then also the clinical trial office here at the Charité, and we have not yet assigned a company for the monitoring and pharmacovigilance, but this will be done as soon as we have more clarity about when we can actually start. Yeah, that's it. Open to questions.
Emma want to start with a question I have heard.
Yeah, thank you very much, Laila. Fantastic. Of course, I'm very happy that we now move to liver transplantation. Perhaps I missed something. I didn't see any biopsies as inclusion or as follow-up criteria. From all the weaning trials we have so far, they are all with inclusion biopsy due to the recommendations of the BANF group. And if you do this, then you basically see it's not 10 to 15%. The spontaneous operational tolerance rate is 2%, because the ones which are in those trials are highly, highly pre-selected for clean biopsies. Yeah. And 40 to 70% are excluded by biopsies showing interface hepatitis, vascular and biliary lesions.
Yeah, fair point. So actually in our case, the biopsy is hidden behind stable liver function. So in our protocol, we state that we need some kind of biopsy analysis in the past year before inclusion, which we consider then as a stable liver function. And this is why we have not explicitly mentioned it, or I have skipped it here for the presentation. Of course, we will not address those problematic patients in this first indication trial here for liver transplantation with our product. We have to see then in further upcoming trials how to handle those maybe more problematic patients.
And the BANF biopsies, have the BANF inclusion criteria for weaning trials?
Emma, shortly, what the colleagues in the Department of Surgery are doing over, I think, 20 years now is that they have routinely protocol biopsies in place, which allow us not only to have the biopsy at the time we want to include the patient, also that we can analyze the course regarding the histology profile, over, let's say, 2 to 3 years. Yeah, and can investigate this, and this is what we are doing before we put the patient on such kind of trial. Yeah.
And also for the outcome, is biopsy also an outcome? Because as you might know, the LIFT and the optimal trial.
Of course, week 60 is a biopsy routinely planned, and of course, if you see any features which are clinically or biochemical relevant, then the biopsy will taken, and we make based on that a decision.
Okay.
Yeah, I have a question here. There was this paper from Qixi Tang in Science Translational Medicine a couple of years ago on liver transplant, similar protocol in the sense of infusion of Tregs several years after transplantation. And the basic message was that it was difficult to produce the Tregs, although they have a particular method to produce donor-specific Tregs, but the message was immunosuppression or something did that there were very low numbers of donor-specific Tregs. So I wonder whether you have run some pilot studies to amplify Tregs from these patients, or you have some thoughts on this?
Yeah, so actually we have done it from from kidney transplant patients, which are on a higher dose of immunosuppression. And yeah, even with a triple immunosuppressive therapy, we can successfully apply our protocol. So we are quite positive that with a very low dose of tacrolimus, we can
But maybe liver has some particularity.
Yes, maybe we cannot exclude this. We don't have liver transplant recipients for the moment. Yeah.
This is on the plan because we have time due to the regulatory aspects, and that is what we want to look up. But it is not done yet specifically, but as Laila mentioned, yeah, we're pretty sure that it will be possible.
Thank you, Laila. Just a quick question. Unlike kidney patients, these patients are already 2 or 3 years on immunosuppression, so they are quite knowledgeable about their condition. So it worth it maybe to talk to them. So the question is whether you did that, because at the Charité, I think there are a few thousand patients who are managed post-transplantation, so you would have the source.
Yeah, so the PI of our study had the outpatient clinic for liver transplantation. And of course, he is in very close exchange with those patients and will already sort out who of the patients may come or may be suitable for the trial, and has talked to them in advance, of course, yeah.
So thank you, Laila. And we continue with you, Carol. Carol? Yes. I already mentioned in the morning this wonderful, let's say, project which comes really up over the whole period of the ReSHAPE, and this is dedicated to CD8 regulatory T cells and all the things regarding manufacturing process and so. And here.
Thank you, Petra. Good afternoon, everyone. Indeed, in contrast to what has been discussed just before and this morning, there has been no CD8 Treg cell therapy trial in the world. So the cell therapy trial, which we will start very soon, is really an important trial. It will be a first worldwide, and this is, of course, thanks to the ReSHAPE consortium and funding that we managed to get there. So if you look at this scheme here, in contrast to CD4s, which you can see at the top of this scheme, for CD8 Treg, it's a long history, but due to the lack of good marker to identify CD8 Treg, it took a little bit of more years, about 10 years, to get to the clinic for the CD8. At the bottom, so you can see that although Treg CD8 suppressor cells were described very early on, they started to be transferred to the clinic only a couple of years ago. And thanks to the work that we have done, but also work from other teams, we managed to set up a protocol for this cell therapy trial. We think that in the context of transplantation, CD8 Tregs have good advantages because they are able to recognize MHC class 1, which is expressed by the graft cell, in contrast to CD4 Tregs, for which, as you know, you can gain more specificity and efficacy if you put an HLA-A2 CAR within CD4 Tregs. So we think that this gives a good advantage for CD8 Tregs, and this is why this is an important argumentation for bringing them to a kidney transplantation cell therapy trial. So we are working in the team since many, many years on those CD8 Tregs, looking for good marker to identify the CD8 Tregs. And of course, we looked at the expression of FOXP3 within CD8 and also CD4 Tregs. And what we were able to demonstrate is that within the CD45 negative population, all of the FOXP3 cells are contained for both CD8 and CD4. So we decided to use that marker in absence of other additional marker at that time, to test the suppressive potential of those cells, which you can see there in red. And you can see that if you eliminate the CD45 high cells, which are mostly Th1 T conventional cells, then you are able to observe a dose-dependent suppressive activity with the CD8, CD45 low negative cells. And we went a bit further, looking at the methylation status of the FOXP3 locus. And we compared to CD4 Tregs, which you can see in blue at the bottom. And you can see that for CD8, the situation is quite close to the CD4 Tregs, although the demethylation of this locus is not exactly as it is for CD4 Tregs. And so one aspect that I'm going to show you is also how we can stabilize the expression of FOXP3 for the CD8 Tregs. So we developed a protocol, so starting from blood from healthy volunteers, isolating PBMCs, and we developed a process ex vivo where we could amplify the CD8 Tregs to gain a population of pure FOXP3 CD8 Tregs. We identify CD3 and CD28 antibody, as well as IL-2, IL-15, and the presence of rapamycin as being critical to get the presence of this population. We demonstrated a couple of years ago that they do amplify very well, comparable to CD4 Tregs, but their phenotype is a little bit different compared to CD4 Tregs. For some of the marker, for example, so FOXP3, although it is well positive, as you can see there at the bottom, you can see that for CD4, if you look really at the high expression of FOXP3, there's some difference of expression in terms of MFI of FOXP3. And we demonstrated that those amplify CD8 Tregs, they do express some suppressive molecules which are part of their mechanism of action, such as TGF-beta, IL-10, and IL-34. And so we showed that if you infuse those cells into NSG mice transplanted with human skin, and similar in the graft versus host disease model, you can have a dose-dependent inhibition of the rejection of the graft, and you can have a dose-dependent inhibition of the GVHD. So we made additional proof of concept, which I'm not going to show here today. In rat and in mice, we demonstrated that cell therapy using CD8 Treg is able to inhibit solid organ transplantation rejection and multiple sclerosis. And we demonstrated in vitro, looking at naive T effector cells and memory T effector cells, that CD8 Tregs, they do inhibit very well the proliferation of both naive and memory T effector cells. And we also published, as Petra demonstrated, showed this morning, that you can also engineer those cells with HLA-A2 CAR. In terms of safety, in vivo and ex vivo, we demonstrated that we can obtain a donor-specific tolerance, and it's not a generalized immunosuppression. We demonstrated absence of cytotoxicity in vitro and also in vivo in the NSG mice. And we also demonstrated that the cells, they do persist up to 80 days in the NSG mice. So with this protocol, which was a research-grade protocol, we moved to a cell manufacturing process. And for this, we moved to the cell therapy unit of the Nantes University Hospital. And we acquired 2 equipment required to isolate the CD8 T-Rex, so the Clinemax system from Miltenyi, and also the Taito cell sorter also from Miltenyi. And we set up a protocol where we could isolate a population of CD8 positive, CD45 versus low negative, which you can see there. Following these 2 steps with the Clinemax and the Taito, which is quite a pure cell therapy, cell product, sorry. And then we develop a GMP-grade protocol to expand those cells into conditions which would be suitable for treatment of a patient. So we changed the medium and the sera, so we moved to the ex vivo 15 GMP culture medium. With human serum 10%, we kept the IL-2 and IL-15, which you can see are used at low dose, and rapamycin. With restimulation of, during these 21 days of expansion, we do a restimulation with the anti-CD3, anti-CD28 antibody. And at the end of this process, we do a conditioning of a cell therapy product in human albumin 4%. We establish a number of release criteria, such as the cell count per dose. You can see in this graph that we were able to reach the high dose for 2 out of the 3 runs, which we did in the GMP facility, but we identify the technical issue which limited the expansion of the cell therapy product. So we are very confident that we can reach at least the 2 first dose, which will be used in the trial, and also the highest dose. We also demonstrated identify the viability, the almost 100% identity of the cells. We added some phenotypic criteria, such as jitter and absence of expression of CD127 into our cell therapy product. And we demonstrated also the function of the cells, and also, most importantly, the absence of cytotoxicity of the cells. Following conditioning, we were able to show that the cell therapy product remains stable for up to 4 hours before administration to the patient. We validated the karyotype of the cells, and we demonstrated that this cell therapy preparation is feasible in kidney failure patients. We checked the methylation profile for cell therapy product, and we were able to demonstrate that 14 or 21 days of expansion didn't change the methylation profile, as well as using cells from healthy volunteers or renal failure patients. We also look at the resistance for a cell therapy product to the different immunosuppressive drug, which will be given to the patient, and demonstrated that there is almost no impact of immunosuppressive drug on our cell therapy product. And we validated once again the absence of cytotoxicity of our cell therapy product in NSG mice. So with this, we are about to start a phase 1/2A clinical trial, evaluating the safety and hints of efficacy of this CD8 Treg in kidney transplanted patients. So it will be autologous therapy, first in human, from a living donor. There will be 3 escalating doses, 1 million, 4 million, and 8 million. The patient will remain under classical maintenance immunosuppressive drug, but this drug can be tapered down accordingly to the clinical follow-up. The CD8 Treg will be administered 1 day before the transplantation. And of course, we will look at a number of primary and secondary objectives, such as the persistence of the infused cells, the migration to the graft, the immune response against the donor, and the conversion of the environment of the cells. And so there will be 2 biopsies, one at month 1 and one at month 3, and we will do spatial transcriptomic to look at the CD8 Tregs within those biopsies, and we expect to see CD8 positive, FOXP3 positive cells within the kidney graft. And so beside this trial, which is about to start, we are also exploring the next generation of the ReSHAPE allogenic cellular therapy. Of course, autologous therapy is very good in terms of histocompatibility, but the patient requires a sufficient number of functional Treg, and this is expensive and slow to produce. So what would be good is ready-to-use, faster, cheaper, and functional Treg. And during this ReSHAPE program, we try to answer a number of questions, such as the capacity, the impact of the cell on the allogenic cells, and whether they would be rejected. And we obtain an additional funding thanks to this preliminary work. So first, we were able to demonstrate that both syngenic and allogenic CD8 Treg in vitro are able to inhibit in a dose-dependent manner the proliferation of CD4 effector cells. We demonstrated also something similar in vivo, but we are currently looking a little bit further into those in vivo data. Of course, they do express HLA class 2 and HLA class 1, so we genetically modify our CD8 Treg using sgRNA and Cas9, and we targeted C2TA and beta-2 microglobulin. And by doing that, with this CRISPR, as you can see there, we were able to get a very nice population of double negative HLA CD8 Treg. And we could demonstrate that those genetically modified Treg are suppressive and able to inhibit proliferation of CD4 and CD8 T conventional cells, and that they expand similarly as the non-modified CD8 Treg. Of course, the non-modified CD8 Treg are immunogenic, as you can see there. So this is looking at the proliferation of CD8 T conventional and CD4 T conventional cells using the CD8 Treg as a stimulator of those cells. You can see that at low ratio of CD8 Treg, there is a proliferation of the T conventional cells, but that already at the high ratio, the suppressive capacity of the CD8 Treg themselves is able already to control this proliferation. But still, since in the clinic we will move with low number of cells, you can see that genetic modification, knockout of class 1 and class 2 really allowed to have a CD8 Treg product which does not trigger T conventional cell proliferation. Due to the absence of MHC class 1, as you know very well, cells can be lysed by NK cells. So first we test the susceptibility of our knockout cells to NK cells apoptosis, as you can see there, in absence of beta-2M. Of course, the CD8 Treg become more susceptible to NK cell lysis. So we genetically modified our CD8 product to protect them using the HLA-E transgene, which, as you can see there, decrease the lysis mediated by the NK cells on the CD8 Tregs. And finally, we looked at the ectopic expression of FOXP3 with the goal to prevent destabilization of this product in a pro-inflammatory environment. As I mentioned before, there is a very good expression of FOXP3, but the level of expression remains low compared to what can be observed for CD4 Tregs. And also, the locus is not completely demethylated as it is observed for CD4 Treg. So we did different things. First, we transduced our CD8 Treg cells with FOXP3, and then we challenged the cells in an inflammatory environment. So first, to see whether inflammation can destabilize our CD8 Treg product in absence of FOXP3, we did RNA-seq analysis, and we were able to demonstrate that a number of genes were dysregulated in presence of an inflammatory environment. What we could see is that when we transduce with FOXP3 or CD8 product, then you can see that there is less gene modified by the inflammatory environment demonstrating that FOXP3 had really stabilized the CD8 Treg product. And in this suppressive assay, you can see that the FOXP3 GFP transduced CD8 Tregs are more suppressive at low ratio of Treg compared to the non-transduced CD8 Treg. And finally, to finish, we looked at, we wanted to understand better how FOXP3 is regulated in CD8 Treg. So using RNA-seq analysis and literature, we identified some genes which we believed are involved in FOXP3 expression and stabilization in CD8 Tregs. And we tested the transduction of a number of these genes to demonstrate how they do impact FOXP3 expression within CD8 Treg. So in conclusion, we generated an autologous CD8 Treg cell therapy, which is ready to use for a first-in-human trial in kidney transplantation. The GMP manufacturing has been successfully transferred into the GMP facility. The protocol was approved by the Nantes Biotherapy Committee, and it was submitted, the CTA was submitted in July 2024. Approved by the Ethics Committee, and we are currently awaiting the final approval from the French regulatory agency this month or beginning of January next year. We already identified the first patient which could be treated with the cell therapy product, so we expect to recruit the first patient in January 2025. And in parallel, as I mentioned, we are working on the modified allogeneic CD8 Treg cell therapy to bring this product to the clinic. And so to finish, I would like to acknowledge all the people who participated to this study. And thank you for your attention.
Yes, thank you, Carol. I'm happy that we got this exciting project running, and really great job.
Thank you, Petra.
Yeah, really. So, questions? Yes. So, Sebastian, first.
Just quickly, do you have funding for the autologous trial?
Yes, so we managed to raise funding last year from the French agency. So it's a ANR DGOS program, which funds the full clinical study.
Great.
So.
Maybe I missed one point. Like during the sorting of the CD8 Tregs, you use CD8 and CD45 RC.
Yes.
And but I see you use the marker for GITA and CD127 as the purity assess. And why you didn't include those two markers for the sorting?
Yeah, we know that these markers, they are expressed by the expanded Treg cell therapy product. And so they are not needed for the sorting, which is already very complicated with the Type2 cell sorter. So this is more related to the function and to validate that the product which we obtain after 3 weeks of culture is really a suppressive product, which we know in at least NSG mice and in vitro as a good suppressive activity. So it's more criteria to validate, like FOXP3, for example. It's not criteria that we use to isolate the cells, but rather to validate that the cells are functional.
So, urgent question?
Yeah, thank you.
No. Okay, good. I would suggest that we make now a break and continue with the two which are left in 20 minutes, that we can have a bio break, a coffee, and all the things like that. Yeah? So thank you again, and we meet us in 20 minutes. Yeah? 20. 2-0. And what the status is so far. To give a short overview, can I have the first slide, please? So this is a Treg Tacrus trial I already mentioned. Here we have the first in class and of course then first in human study. for investigating safety tolerability in a phase 1/2a of the gene-edited regulatory T cells, which are tacrolimus resistant as an adjunctive therapy in living transplant patients. So we have, as you see, refer to the, again and again, to the one study trial we have done in the one study consortium. And I already mentioned that regulatory T cells, of course, sensitive to concurrent immunosuppression, and Carol showed very nicely that the sensitivity to different immunosuppression for the Tregs is different, but nevertheless, the tacrolimus also have an impact on the functionality of the cells, and even more. If we go a part of the solid organ transplantation and want to go towards autoimmunity, it is essential to figure out a strategy in which you can combine the regulatory T cell approach with classical immunosuppressive drug to control the effector T cells efficiently and give the regulatory T cells an advantage. For any kind of gene editing, it is important to have a really close eye regarding safety on the purity of the starting material. Because what you have to prevent, of course, is that you just by chance gene edit some of your impurities. And even if I show you that we have in our product impurities, let's say, no B cells, no NK cells, but some CD8s, of course, and CD4, CD8 double positive cells and something like this, around 1%. So this 1%, if it is resistant against the immunosuppression, is a danger. Yeah, they are uncontrolled. And therefore we spent a lot of effort. I will show you in the next slide on figuring out a process and develop it and get it approved to have really a pure as possible starting material for all the other procedures. So I already highlight that we come up at the end also here of a process with a customized single guide RNA in combination with Cas9 to achieve a high knockout efficiency dependent on which technology you are used for figuring out the kind of efficiency. We are close to 100%, for sure around 90%, but for good reasons we decided to have at least a threshold of 70% of knockouts in our product, because you will see it, this is a part now of our release criteria, and it is a must what we have to fulfill. Again, I will not highlight, but here also the process start from 50 ml peripheral blood. So, and based on the isolation and the purity I here mentioned, and this means that we have an impurity which is below 0.1 and even lower, yeah, as a starting material. You will see that based on a new sorting strategy, we come up with a more tiny starting cell number. Also for the classical polyclonal expanded Tregs out of 50 ml, we have a range from diseased patients, yeah, not from healthies, which range between 2.5 to 4 million. Andy was saying so. If we now use a new strategy, which I show shortly here, then we have 500,000 to 1 million, so something like this. So, and again, we stick on the same protocol. I don't change something except the kind of isolation procedure. The normal procedure for regulatory T cells is that you have first a depletion step via Clinimax. You take out the CD8s and you enrich for the CD25, and then you expand them. So again, the impurity is an issue, and we try several strategies. Start with kicking out the CD8s, enrich the CD4s, and then sort with a Taito, which is also a difficult machine for the 127 negative. This is not feasible because we cannot identify the purity based on identical CD25 monoclonals for detection via the flow cytometry. Then we do 2 times the Taito. It was difficult because we had to start with the Ficol preparation, which is not really GMP compliant, I have to say. Rosetta Zept, that is what I learned, surprisingly, is accepted based on, obviously, the history of 30 years ago. But I feel not comfortable. This could be, and you can achieve high purity of the cells you want to have as a starting material. At the end, what we come up is a double positive sorting strategy with we enrich CD4 by Clinimax, and then we sort the enriched CD4s with a Taito. So here you see the purity lower than 0.1% of the CD8s, no NK, no B cells, which is important. The second step is the gene editing. And I already mentioned that this is a release criteria now, and therefore we have to define what is a worst case scenario we want to achieve, because we had to answer, of course, at the end, what is or how can we explain the result in relation to the gene-edited cells. So, and therefore, you have to know how many you have in. What was here a challenge, believe it or not, at least in our hand, we have to have all reagents which we are using has to be GMP compliant. So it is. So that means it took a little bit time to get the customized single guide RNA produced. And also to have a number in place. It was not only a matter of time; it was also a matter of money. Because the smallest amount of single guide RNA generated GMP compliant cost me 350,000 euro. So, yeah, only so to understand why I'm annoying, because it comes. So we receive it in July 22. And I was in a hurry because I want to complete the manufacturing process and start with a clinical trial. So therefore, Madame decided that we have now the single guide RNA in place. We know that we have no really differences regarding the non-GMP compliant Cas9 in comparison to the GMP compliant Cas9. Yeah? And I decided, stupidly, I have to say, that we make the validation runs to have the results in place, which I then hand over to the authorities to say, look, we achieve what we want to have, and please give us the manufacturing authorization. And this was not the case. We had to do first a comparison between the non-GMP grade Cas9 and the GMP grade Cas9. And furthermore, we had now the problem that the single guide RNA was already there over 2 years. And that means we had to start with stability testing of the starting material, because it is a starting material. And so far, no really design is given by the regulatory authorities. That means we had to develop our own stability testing program for the already stored material and have to implement this in our protocols. So, yeah. Oh, next one. And the next one is the product specification and release. I already mentioned that we use a whole portfolio of technologies written here, epigenetic, single cells, site-seq, and all this so fancy stuff. Dieter already show you what comes out. We are fine with product characteristic, and based on that, we come up with this as a release criteria. And you see that beside of all more or less well-known things regarding viability and cell composition and cytokine producers and beat depletion, we have now to incorporate the exact number of cells who are genetically modified. So, next one is that we approved it, that we get the manufacturing authorization approved. Yeah, so. And this is a protocol, very tough and very easy. And we decided to make it more or less one-to-one, for good reasons, I hope, in line with our protocol, what we have established or what we have followed in the one study, also meaning we deal with living transplant recipients. We take the blood roughly 2 weeks before we want to we have the transplantation in place. We have time for the manufacturing. By the way, here we need a little bit more time. We have not 16 days, as I mentioned, for the polyclonal product. We need here 21, correct, Andy? 20, okay, 20, 1 day less. So as a 20, and we will infuse the product exactly at the same day, day 7, because I try and I think that makes sense, that we compare the results from the one study, from the polyclonal expanded product, in all aspects with a new one, and that means that we have really the possibility to have the one-to-one comparison, also based on the study design. So, this is a dose escalation. We go forward. You see that that we have here 4 steps of dose escalation, because I believe that based on the higher purity and on the more stable TSDR demethylation of the cells, we have a better functioning product in place. And based also on the one study experience that we don't see any any comparison which are dose related to the efficacy and neither to the safety. I was a little bit, I had concerns regarding safety, and therefore we start with 0.25 million cells, and then we continue with the same dose escalation as we have it in the one study, that at the end we have the possibility to come up with all the more or less good clue about the dosage in this content. So this is our achievement. We have achieved the manufacturing authorization for the starting material I already mentioned. We have qualified our GMP facility, which was primarily not designed, but not as I start with cell and gene therapy, I was not really willing to come up with gene editing approaches. So that means we had only an S1 approval, and now we have an S2 status. Of course, we have done the validation runs, we have the manufacturing authorization, we got inspections, we have now the manufacturing authorization for the gene-edited product. We have submitted the clinical trial application. And based on all these efforts, what we have achieved, we come up with another idea. And this is that we apply by the EMA Science Innovation Office for, let's say, a support regarding all regulatory issues, study planning and implementation, because they realize now that it is in purpose and that is an issue, and that academia and also non-profit developers suffer from some obstacles. And the idea was or is that we want to implement really the so-called basket trial design, which is firstly accepted by the FDA in 2018 for monoclonal antibody for cancer treatment. Now we want to adapt this as a role model for all of you guys later on for the non-cancer field. This means again that we we were successful, as you see, together with Teleton, we get green light, but after demonstrating the first positive results, of course, of the phase 1/2A. Therefore, I'm a little bit in a hurry. regarding this. And the aim is, of course, based on our expertise so that we first try to implement a basket trial design for all transplant, solid organ transplant settings, and then translate this towards autoimmunopathy regarding eligibility criteria, a definition of subgroups, intervention assignment, but more importantly, and therefore I'm really proud that we have done so much effort regarding the biomarker portfolio, because we have to demonstrate, if we go for such trial, not only the biological plausibility, we have to go for accuracy of the biomarker tests. And accuracy of the biomarker test means in this case, which makes sense, to figure out the false positive rate to avoid an underpowering of the trials in this sense. And this is a little bit a difference if you have to figure out the false positive rate in an appropriate GCLP conform manner. And the second point is the sample collection has to be, what is written here, should be easy. Yeah, should be easy and relatively uniform. So regarding quality and also regarding the yield, which has to be then analyzed. So that's it from my side. And again, my thank goes really deeply to all my colleagues showing here from the GMP team, from the TREAT team, which take this over and adapt this strategy. It's not surprising, Laila is part of the game from the beginning on towards effector T cells. Again, I thank Robert Oellinger for taking over the PI position, Brigitta Globke as co-PI, Cyrent. All the data which I don't present because Dieter have done it regarding the single cell analysis were done, not all of them, but many of them done by Fasim Mashregi, so from the German Rheumatism Center, and yeah, Shaggy Moon I mentioned several times, now it's enough. So, questions? No. No. Questions?
Hi. I saw the expansion protocol where it mentioned the semi-closed system, and I see a G-Rex picture. How does that work with the German regulatory authorities? Is this good? Okay.
This was not an issue because we adapt, as I would say, any 8 years or something like this, all processes to the G-Rex, which is per definition, and at least in my eye, not a real closed system, but it is dedicated to be a closed system. So this was fortunately not an issue.
Thanks.
So, Dirk?
Thanks. Well, it's of course a very exciting study. I was wondering, looking at the protocol, if you decided to introduce the modified T-Rex early on, day 7, and if I saw the sort of timeline for the immunosuppressive therapy, it's of 36 weeks or something. That
yes.
It's going to be reduced, what safety measures are in place that, let's say, it works very well?
Mm-hmm.
So you enter your patient actually in a state of even over immunosuppression before you can start to reduce the classical immunosuppression. And so my question is, what specific safety biomarkers are in place to cover that period?
All these biomarkers, I prevent to repeat myself, all biomarkers which we have implemented in the ProTrack, which we have implemented in the one study. Also all these safety biomarkers are in place. And again, the point is, Abul asked, is it really necessary? And yeah, I cannot answer it so. Is it really necessary? Because with a polyclonal expanded Treg product, we are to many extent fine. Yeah, we have a good long-term outcome. We could achieve, let's say, 5, 6, 7 year monotherapy safe. So, is it really necessary? Is the product really better than the polyclonal one? This is what we want to answer, because it is also one important point for, let's say, the next strategies. Do we need it, such kind of procedure, only because we believe it? So, therefore, again, I think from my perspective, it is wise to have the really one-to-one comparison in all aspects. And in the one study, indeed, we treat the patient at day 7 under the whole umbrella of immunosuppression. And yeah, what we have achieved, at least clinically, is repetitively demonstrated, I think, from both groups. So, yeah, this is maybe the answer, and we look for viruses, we look for all the things, for, let's say, the change in the TCR, but also all the fancy things. Yeah? Okay, good. Good. So, now I'm really proud, I have to say, that two enthusiastic people, maybe colleagues, not really, but yeah, persons who take part of another EU consortium, Gene Tiger, we have in place, and we had yesterday and the day before our retreat here as well. Juan and Monica are part of the Gene Tiger consortium, and they are patient representative. I ask him, invite both of them to give us a little bit patient perspective on all the things we are here doing, and which question we have to take in account that the patient organization is well informed, that we understand each other, and that we go in the more or less good direction to regulatory authorities to convince them that you are waiting from of what we here try to implement. Yeah, thank you for both for coming. Yeah, I'm really happy.
No, thank you, Petra, for the invitation. For us, it's a very good opportunity to spread our vision about how entities like us, how patient organization could collaborate in clinical research, and with the new regulation, the new HTA regulation, of course we have a new role in this way. Following the title of the presentation, I will start talking about how cell and gene therapies suppose for patients in general. First of all, of course, the first hope is to cure the disease that produces kidney failure. It's the hope of all patients, not only for kidney patients, but it's the first hope, of course. It's the primary aspiration, and because chronic conditions are really difficult to offer good treatments in this moment. For this reason, the second hope, this is depends of the personal situation of patient, but also the time, for instance, the time for diagnosis, for instance. The second hope is life expectancy to offer survival. For individuals with chronic condition like kidney disease, extend the life expectancy is a new hope. Is not possible a cure of the disease, of course. And the third, for us, is to enhance quality of life, because as not possible at the moment, the cure of the disease, to reduce the side effects of other issues that the treatments have, enhanced quality of life is really important for us. In summary, the hope surrounding CGT is multidimensional. It's not only not just important the clinical data, it's also important what's happened, what is the impact in the daily life of the people, of the patient, and what's happened with their life, because in disease like kidney disease, it's not possible a cure of the disease, but it's possible to extend a lot the survival. Of course, this is the hopes, but there are many concerns about this kind of therapies. It's different if we are talking about individual patients or patient organizations. And regarding patient organization, this is our concerns. The first is the high cost that this kind of treatment have in general. We expected high cost of CGT treatment and represents a significant concern for patient organization. And following these concerns is the second, the universal access to this treatment, especially in countries of low incomes, not only, but in countries of low incomes. And the third is the possibility of health tourism in the same direction. This is the third concern that we have regarding this kind of innovations. Addressing these concerns will require collaboration and effort of many, many stakeholders, not only patient organizations, even regulatory authorities, EU Commission, and you, scientists and professionals. And to explain the next, I give the floor to Monica to explain the expectations.
Thank you. So, well, as Juan Carlos was saying, we also have expectations regarding these CGTs. We've divided it like in three. So for researchers, well, or from the point of view of us regarding researchers, it generates high expectations for the ability to transform a treatment. Also regarding regulatory authorities, they mark critical milestone and they enable or not this access in Europe. And also the new HTA regulation that Juan Carlos mentioned that can have a patient-centered perspective to the decision making. The expectations we have toward clinical scientists is to involve patients from the beginning of the research so that we ensure that patient perspective is included. And also it's to shape the goals of the studies. Towards regulatory agencies, the expectation is that they focus on safety and efficacy to ensure that these therapies are both effective and safe for patients. And towards EU Commission, in this case, to ensure that they inform patients clearly and effectively. Regardless of the background or the location, we know that's quite challenging right now, but at least it's the expectations. We want to talk about why patient participation is important. We know sometimes we don't have that much scientific data to show why this is important, but patients are active agents and they have a lot of insights and their participation is really meaningful in research. It ensures that studies are more applicable and that they're relevant, that are conducted with patients and not only for them, and it improves the impact that the study could have. We also are seeing a new profile of patients. In general, they are more informed, more engaged, and they are also more demanding. So they seek to contribute also to the development of some of the solutions. And we know that involving patients have some barriers and challenges. We know there's a lack of awareness. Usually we don't really know what the benefits of having patients involved are, and sometimes are underestimated. We know there's limited evidence on the impact it may have. So, of course, measuring this kind of collaborations with the standardized scientific criteria, it's still a challenge. There's, of course, insufficient training for patients, but also for professionals, since we lack the adequate training to collaborate together. There may be, of course, language difficulties, not only talking about language itself, but sometimes the way we express ourselves is not easy for the collaboration. This is also, well, a different topic is the time burden for patients that are being asked more and more to be involved. So this is an ethic and compromised issue. And also there's this institutional resistance to involve patients, as it's usually viewed like a requirement for some things, but it's not really more than that. So incorporating the patient perspective means that you have actual people, the actual individual that has lived with this disease, so they have a lot to say. Also that they may have different priorities. Juan Carlos will explain later a bit more in detail, but maybe the concerns that they have may differ from those of scientific professionals or healthcare professionals in general. The project may have an improved clinical relevance, as they may ensure that the questions asked or the hypotheses are the right ones and are the ones that the public are asking themselves. It also increases the trust in research. So it's it's brought to patients and general public, and it may have better outcomes and a better use of resources, which is always something very, very important for all of us. Now I'm going to give the floor again to Juan Carlos to talk about the patient perspective in a scientific way.
Thank you, Monica. Okay, to finalize our presentation, I would like to explain how we can measure the patient perspective. Because if possible, the scientific measure of the patient perspective, yes, it's possible, through, for instance, psychometric tools. The typical answer to this question is, well, we can, when you put into practice your achievement in this kind of project, you can measure together your clinical data, shown measured from the patient perspective. And the usual answer is, we can measure the quality of life. Quality of life is the PROM, the patient-reported outcome measure, more usual in clinical trials, and little by little more usual, and little by little more interesting for regulatory authorities. But regarding kidney patients, there are some problems about the generic quality of data, or the quality of life tests. For instance, we used three, the most usual health-related tools in kidney patients, and we found differences between the three tests. We use Eurocool, SF36, and Hue3, and this is the curves of the results, and you can find differences between the curves. And what does it mean? Probably this means that different quality of life tests are measuring with different precision different factors of the quality of life. And this is important in order to compare studies and compare group of patients in different trials. But quality of life is the only measure that we need in order to address, in order to capture the patient perspective. Well, one of the problems is this, or other study that we promoted, is that quality of life are affected for other factors. Very important, for instance, the emotional disorders. Why? Because we found that quality of life tests are not good tools to measure the emotional state of the patient. And in the case of kidney patients, one of the most common symptoms of kidney disease is anxiety, for instance. And anxiety is underestimated, under-diagnosis, and under-valuable in quality of life test. It's one of the problems. Why happens this kind of questioning in kidney disease, at least in kidney disease? One of the possibilities is because the perspective of patient and expert are different in different dimensions of the healthcare, in the field of healthcare. For instance, in this study, we requested expert and patient, expert CRO, the professional that advise research, we requested to rate different dimensions in the field of health. This is the blue line at the professional responders, and the red line are patients. And we have a very important agreement in some dimension around about physical, affective, and cognitive dimensions. These are the dimensions that quality of life tests measure. But we found very important differences about satisfaction and experiences. Usually, this kind of dimensions are out of the quality of life test. This is important because if we are trying to analyze the impact in the daily life, the impact in patients about your research, you are losing very important data. In other study, this is our last study, in the left, the table of the left is the agreement between experts about the dimension of a specific test that we are developing for AUS, for atypic hemolytic uremic syndrome. And we found a very important agreement. This is the index is up to almost 0.6. A very important agreement between experts about the items defined for the questionnaire. The same for patients, very important. However, when we asked patients and professionals about what these items measure, if the item measure experience or outcomes, patients have a total agreement that all the items measure experience or outcomes. However, the professional responded that some of the items didn't measure experience or outcomes. It's the same that the previous study. There are dimensions that are important for patients, but are missing for professionals. And this is important because most of the tools that we have to measure the patient perspective are developed only for professionals, for healthcare professionals, without the perspective of patients. And our study shown that there are dimensions that are important for patients that should be in this kind of tools. Well, this is our summary because there are other studies that shown that it's really important to put the patient from the beginning, not only in the develop of this kind of tools, even in the design of research and all of the situation that needs the participation of patients in this project. Thank you so much for your attention.
Again, I thank you both for your participation, for your active involvement in everything what we are doing. Indeed, I strongly believe that we need you, not only as a target, let's say it in this sense, we need you really on our side because at the end, we want to make something better for our patients. That is our intrinsic point. So, do you have any questions, Dieter?
Yeah, thank you. Also from my side, I have only one remark. I think the one issue, particularly in Europe, is hurdles of regulatory authorities, particularly to come with new things. And what I miss, in contrast to the rare disease community, the patient community, that there is not, the regulatory authority interaction is not on your agenda. And I think it's a very important point that you also bring your view to this, and we see it have impact. For example, rare diseases, you have many new or easier access opportunities now by regulation. And I think here you have a chance because sometimes the physicians, they don't believe they are too much straightforward, but maybe they believe you. and I think you could have an impact on this, and you should put it on your agenda as well.
No, for sure. Rare diseases, rare kidney diseases is really important for us. One of the studies that I saw is based in what's happened with house patient that is a rare kidney disease. But of course, it's something that is in our agenda. In our federation, we have a specific federation for rare and genetic diseases that is federal.
The regulators, right? I think you're asking about that we didn't include like, sorry, Petra.
But this is our way to show regulators that this kind of treatment could be interesting for our population. Yeah. Not only about clinical data, also about the patient perspective.
A question from Sebastian.
So the whole thing is about the patient journey, isn't it? I mean, this is unusual about the immunosuppression trials because it's always the patient's choice. You get told before this whole thing starts, your life will change. You need to be on immunosuppression, and that has all of these risks. And then someone tells you, and now we might reduce the immunosuppression, maybe. And some patients will have a completely different reaction to that than others. I was hoping when these projects started that we could have some empirical social science research linked to that experience to capture those patients' experiences who's like, well, I'm either going to take part in this trial or I'm going to stop taking my drugs anyway because I don't want to be on immunosuppression for like that. And it's, with respect, it's not about rare diseases, especially if they're doing a basket trial now. It's about the entirety of that experience that could inform other disease functions as well. So I think that's maybe where the impact or the feedback could be most usefully applied, because it's not something that clinicians necessarily recognize, and regulators certainly don't.
No, that is a very important point, because one of the question is not only the emotional state affect kidney disease. It's really important to control people with a disorder in order to detect what is this group in order to offer a real effect of the treatment. I mean, this is the question. For this reason, the combination between outcomes and experience is really important. And also to analyze if your group has a high level of emotional disorder or not, in order to control them.
So, thank you again. Sorry. Oh.
Thank you very much. Just coming back to the point about the regulator, actually quite a few studies have shown that patients have a much higher risk tolerance than clinicians and regulators.
Yeah.
Actually, patients want to go into trials and they want to try new things, and clinicians are risk averse, and the regulators are even more risk averse. No one wants to take responsibility, not even for the blood tube that we're taking the blood with. You know, no one wants to take the slightest bit of responsibility. The way around that is for patients like you to write to the regulators. So in the FDA in America allow patients to write to them to say, actually, we really want this. So I think that's what you should be organizing yourselves to do in Europe. You should be writing to the regulators.
We are already reaching regulators, but I don't think we have as much power as you think we do.
No, that you
But
have the power.
Yeah, it's true.
Yeah.
With your perspective so that the regulator will take it in account, and as more as we come together, that is the point. As maybe faster we can achieve what we want to investigate, and we take the risk, you take the risk as a patient, because you trust us, and we have to say, we thank you, but we cannot really say that you will not be suffer from any kind of treatment-associated issues, sometimes also severe one, which means this is indeed an issue, and I'm pretty sure that the authorities will not ignore it and will take it as an also chance for them to be more flexible in many decision points. So last point, because...
Yeah, I just wanted to echo what Vadim, Dieter was saying and what you've just said, Petra. I think it's really wonderful to hear your perspective, and thank you very much. And I think it's exceptional that you've been able to produce such interesting and clearly quite rigorous and well thought through research on patient experience and outcomes. And you've demonstrated this extraordinary synergy, but equally the disparity between the patients and the healthcare and scientist professionals. Echoing Fadi's point, it would be really interesting, therefore, given that you have this capability to produce this kind of eloquent research, to pinpoint things that would be enormously helpful for scientists and clinicians vis-à-vis the regulatory point. For example, would patients prefer to have research availability on a compassionate use basis versus the rigor of the let's just say the first in human clinical trials? Now, you could actually design that in a very straightforward way that's accessible to patients in such a way that's easy for them to answer. And I think it would be an extraordinarily valuable piece of work that would be taken very seriously by the regulators. And I really urge you to pursue that.
Good. You take it as an duty for the next years. So we continue and we change a little bit the program because Annette Künkele has to take care of the children, I think. But I'm happy to have her here because she coordinates another EU